in India, and then we look forward to. Just to give a two sentence about the topic, particularly those who are students here, undergraduate students. All of you know, of course, genetic material, DNA. And DNA, once it gets, the information is decoded, most of the coding part of the DNA is related to making protein. There are, of course, other parts of the DNA which is not going to code for the protein. But much of that information is used for how much of the protein to make, when and where to make that protein, so-called regulation, spatiotemporal regulation of the protein synthesis. But once the proteins are made, that's really all life is all about. Finally, the structure of your cells, structure of your very different organs, tissues, individuals, our interaction with the environment, how we perceive, everything is mediated through the functions of these proteins. And the proteins have structural function as well as catalytic function. And all of these things depends on the conformation of those proteins. The structure of the protein determines what the function is. The function is an emergent property of the structure of the protein. And the structure of the protein depends on the chemistry of the amino acid, which make long chain of these proteins, polymer of amino acids. But it is a challenging problem. And understanding the life, all aspects of life depends on how you understand the proteins. How we understand the protein depends on how we understand the structure of the proteins and how do they acquire those structures. And there's no one else who could talk about it at length amongst the scientists who are in this in the world today, other than Professor Janet Thornton, who has a tremendous several decades of experience in this area. She has seen the emergence of this field, as an undergraduate student all the way to now as the senior most scientist in this field. And it's really an honor and privilege to hear the protein world from you, Janet.
Thank you.
Thank you. Thank you for coming.
Now I would request Ms. Asha to say a few words about the Shivalal Philanthropic Initiative, please.
Thank you. So greetings, good afternoon to everybody. I'm Asha Thomas. I'm the CEO of this Shivalal Philanthropic Initiatives. And first of all, I'd like to thank everybody at NCBS for inviting me over. You're a beautiful campus, very serene and amazing the amount of work that's been. I didn't know there was so many aspects to biology, by the way. So from the innards of proteins to I think ecology, research to outreach, it's amazing. It was a quick tour, but very enriching. So just to tell you a little bit about the philanthropy, this was started by Mrs. Kumari Shibulal back in 1998. It started as a very small charity kind of thing, and now we are 25 years old as a philanthropy. The overarching goal of the philanthropy is to build equity in society. So we do this by working with underserved communities, in education, skilling, healthcare, organic farming, and preservation of art and culture. Because we are working in equity, we think education is the greatest equalizer, and so 80% of our work is in education, and most of it is school education. School education, college, secondary education, and skilling. So to give you an idea of the scale and the impact, our first flagship project, the Vidyadhan project, working with the scholarship for secondary education, has touched 14,000 students of the past 25 years. We have another program that works with the government and the community. It's a more systemic program, which is both a technology platform and it is also an orchestrator which brings together the NGOs, the government, and other individuals and school leaders. Basically, it is about improving school education, and the school leader is a very important actor in this whole ecosystem. So that is working with about 1.5 million schools today. So this is, in the past 25 years, this is the kind of work that we have done. So apart from this, back in 2000, Mr. Shibulal felt that research in India was very early stages, and he wanted to encourage research in India. And he is an alumnus of TIFR. So in collaboration with TIFR, he brought in this Fellowship, which is now we're calling it the SFPI Fellowship, which is the Shibulal Family Philanthropic Initiatives Fellowship. So that is the origin of this. And as part of that, there are two parts to it. One is it supports young researchers from India to travel abroad and to collaborate with their colleagues abroad. And the second part is where we bring in eminent scientists from across the world to give public lectures in different cities in India. So it is as part of that today we're very happy to welcome Dame Janet Thornton to India. And she's already finished her Mumbai trip, and now she's here in Bangalore. So so welcome, Janet, from SFPI. And also I think all your lectures and interactions will definitely enrich this student community here.
I hope so. Thank you.
Thank you, Asha. So first of all, we are so thrilled and happy and grateful that Shibulal Foundation Philanthropic Initiative is funding this trip by Professor Janet Thornton. And it's my great pleasure to introduce the speaker. She's the Director Emeritus of the EMBL-EBI in Cambridge, UK. After her early education in physics and doing a PhD, she took over as the head of crystallography unit in Birkbeck College back in 1978. And also she'd been serving in the biochemistry, molecular biology department in the University of College London. And later in 2001, she took over as the director of EMBL-EBI, and she was serving for until 2015. Professor Thornton is one of the world's leading researchers in structural bioinformatics, using computational methods to understand proteins, structure, function, evolutionary relationships. She works at the interface of physics, biology, chemistry, and computing. She has made some very early, fundamental, and influential contributions on different areas of protein structures, including disulfide bonds, salt bridges, and beta hairpins, also in protein structure prediction, and she has also been instrumental in presenting tools such as Threader, which is a very important protein structure prediction method, as well as recently to study the divergence of enzyme function. Some of her highly cited works include the database CATH, which stands for Class, Architecture, Topology, and Homology of Protein Folds. Their web server called ProCheck that is popularly used for protein structure validation. It's cited more than 27,000 times. And another tool from their lab, which is called LigPlot, which is used to describe protein-ligand interactions in 2 dimensions in a very conceptual manner. It has been cited 8,000 times. Professor Janet Thornton has totally published more than 500 research articles. She has served in several committees, such as the Protein Data Bank and the BBSRC, and also served in advisory boards of around 20 institutions worldwide. She has won several awards, and it will take me a long time to say all of them, but I'll say Hans Neurath Award and the Extraordinary Fellow Award by Churchill College. At least a dozen universities have offered her honorary degrees. She was elected to the Royal Society in 1999, and she's a foreign associate of the US National Academy of Sciences. In 2012, she became a Dame Commander of the Order of the British Empire, knighted by Her Majesty, the Queen, for services for in bioinformatics. Janet is an inspiring scientist who has been humble and encouraging and showed us that it is important to embrace the fact that certain areas of research tend to be often crowded, and it is also important that friendship has to be preserved more than anything else. She's also an inspiring woman scientist. I understand that part-time research was introduced in the entire history of the science in UK due to Janet.
Wow.
And And I have personally very much got inspired by her great science and the way she has been a woman scientist. As mentioned earlier, this visit by Professor Thornton is supported by the Sarojini Damodaran Fellowship for Academic Visitors of Excellence, funded by the Shibalal Philanthropic Initiative and the Shibalal family with the support from TIFR. She had been in Mumbai during the past 2 days, and she has given a couple of public lectures there already. And she will be giving actually 2 lectures tomorrow in Indian Institute of Science. One is a science lecture in the noon, and then there will be a public lecture in the Biological Science Auditorium at the same time, at 4 p.m. Today, Professor Thornton will be talking about the contributions of bioinformatics over the last 50 years in deciphering structure-function relationships of enzymes and who else can say it better, because she's been in this area since 1974. I hereby invite Professor Thornton to deliver today's lecture. Please.
Thank you. I think so. Yeah. Good afternoon, everyone. It's a real pleasure for me to be here in India. It's even more of a pleasure to be here in Bangalore and to be at the NCBS, because partly because I have known Professor Soudamini since I was a postdoc, and to come here and see your wonderful campus and to hear about all the work that's going on in the Institute really is extremely good. I'd also like to thank the Foundation for funding me to come here. After COVID, I think people have not travelled so much. And in fact, this is the first time I've travelled intercontinentally since, well, not quite, I went to Canada last year. But this opportunity to meet with other scientists is really extremely important, I think, in the development of science. But today, I'm going to give a lecture which is, the first part is historical, to give my personal reflection on the evolution of structural bioinformatics. And then I'll go on and say a little bit about the future and the impact that I think can be achieved in this field. So, historically, one of the greatest, I think, insights came from Linus Pauling when he proposed that protein structures would have alpha helices and beta sheet. This is really insightful because there were no protein structures at that time. And the first protein sequence wasn't determined till 1953. So, given everything we know today, it is quite amazing that it's only happened in the last 70 years. We didn't know a single protein sequence before 1953. The first protein structure was determined in Cambridge in 1958 and followed by for myoglobin that we all know and love, and hemoglobin in 1960 at the Lab of Molecular Biology in Cambridge, UK. I should also point out that Ramachandran, who is your very famous biophysicist in India, had his own insights that I'll go on and mention in 1963. And that, I think, visiting, I also sense his presence in a lot of the research that's happened in India since that time in this area. And it just emphasizes to me the difference that a person can make in research. Since then, in 1965, Margaret Dayhoff, who you can see here, I hope, made the first collection of protein sequences in a book, a pink book. And the pink book used to lie on the desk, and we used to, and it was made by typing in these sequences. I mean, it's amazing. And she brought all these sequences together. That allowed sequence alignments, which are at the very basis of bioinformatics. And after that, since then, we now have many protein structures. But the Ramachandran plot is one of the, to me, it's one of those things that I would have loved to have done. It's really insightful. It's very simple. It just looks at the dipeptide and the change in the conformation for the dipeptide of the protein here, and tells you which are the allowed regions and which are disallowed. And so that basic stereochemistry applies throughout to all proteins and has been very important insight. And you can also see here Dorothy Hodgkin, who was in Oxford when I was there, and Tom Blundell, who I think has been a very frequent visitor to India. And I was in his department at Birkbeck. Of course, we now think of the Ramachandran rather differently, because we've changed the definitions of what the phi-psi angles are since it was first solved. But that's a detail. So, for me, one of the most important things that happened was that in 1971, the Protein Data Bank was established at a meeting at Cold Spring Harbor in the States, when they decided to create a database of these protein structures. I think at that time there were 7 structures that had been determined. And that actually, without the Protein Data Bank, I couldn't have done any of my research. It has really changed everything, and we owe a great gift. It was a real gift to gather this data, and you'll see why. Since then, so I started looking at proteins in 1973, which is about here. There were 20 known protein structures. Today, well, last Thursday actually, we had 230,000 odd experimental protein structures. And so what's happened is that the universe of protein structures has been revealed in my academic lifetime. So it's a bit like finding the structure of the world, when the explorers went to explore the world and they didn't know what they were going to find. And certainly when I was in Oxford during these years, every time a new structure was determined, we would rush to the library to see what this structure looked like, because we didn't know what it might look like. Would there be anything else other than alpha helices and beta sheets? And gradually, over this time, we've come to learn more about these structures. And of course, the key thing that was postulated by others, but by Hinxton mainly, is that the sequence determines this 3-dimensional structure, which is not only extremely beautiful, in my opinion, but it only works because of this structure. If you lose that structure, you have no function. And so structure is a very important aspect of understanding how proteins work. So, what happened? We were trying to, of course, not only did we want to understand, we wanted to predict. So we wanted to give me a sequence, you tell me what the protein structure looks like. So how do you go about that? We were novices, we didn't know. What do you do? And so in the 1970s, when I first started, we tried to tease the problem apart, to look at the different bits of structures and see if we could predict those. So we looked at the geometry of the chain, the phi-psi values I mentioned. We looked at the hydrophobic core and the hydrogen bonds and the disulfide bridges, beloved of many here. The secondary structures, helices, strands, turns, different types of turns. I spent a long time looking at beta turns. And here in India, other people have looked at the alpha turns that were relevant. And we also looked at chirality of these. So we all know that helices are right-handed, twists are left-handed. And that all comes just from the structure of the amino acid. And that fact, again, still amazes me, that this little change between an L and a D form can have this huge impact on protein structure. And of course, the impact of water solvent accessibility. So we studied all of these and we tried to predict them. and with varying results. We actually used neural nets and things to try and do these. We had different methods of doing it, and we found lots of different beta turns. We looked in great detail at how the 20 different side chains interacted with each other. But one of the big challenges was actually looking at these structures, because then what came out of an electron density was a picture something like this. And from that, it was quite difficult to see. Even when you had stereo views so you could see it in 3 dimensions, it was still quite difficult to tease apart exactly the path of the chain. And Jane Richardson developed, she was an artist basically, and she drew these ribbon diagrams by hand. So I went to visit her lab, and she had all these big pictures of proteins that she'd drawn, quite fantastic, that really allowed us to see how the chain folded round and created this globular structure, which could, in this case, bind 2 zincs. This is superoxide dismutase. And then over time, of course, Jane's ideas were captured in a computer program, so we can now look and automatically draw these topology diagrams and see what's going on. But that might make you think that you can see through proteins, and of course you can't see through proteins, they are very solid. And so this view, which I call the CPK, Corey, is an important aspect, that we look and we realize these structures are very solid. As Minnie mentioned, proteins, their interactions with ligands are very important. But looking at 20, 30, 40 structures and 40 ligands and how they interact is very difficult. So we developed this tool, or Roman Laskovsky developed this tool that allows us to see it in 2 dimensions and to compare between different ligands and different proteins. And also the hydrogen bonding diagrams, and these diagrams that show the sequences, the helices and the strands. So this visualization is important. And I also included this from David Goodsell, which is, I think, drawn by hand, that stresses how in a cell, how tightly packed all of these things are. So they aren't like the continents of the world scattered around. They're really close together and interact like that. So these tools to visualize structures were very important. So we had efforts at secondary structure prediction. Chu and Fasman was the one that everybody thought about and used. There were exchange matrices that refer to how amino acid mutations occur and the likely chance of an arginine changing into a glycine, which doesn't happen very often, but it's there. The annotation, the mere annotation of given a structure, tell me where the helices are and the strands. At one stage in the mid-70s, there must have been about 20 programs that did this. And of course, the quality of the secondary structure prediction depended on that annotation. And so if everybody was using different annotations, they all got different values. And so Chris Sander and colleagues, Kabsch, who actually drew the topology diagrams, developed this program DSSP. So these individual programs can be very useful in actually defining where the helices are, and this is now fairly standard. And also, we were trying to predict secondary structure. And what Burkhard Ross did with Chris Sander was to include multiple sequences. And they found that you could significantly, 10% probably, increase the quality of secondary structure prediction using multiple sequence alignment, so that you're averaging, you get rid of some of the noise, basically. Then in the 80s, so that was the 70s, and we only had, I don't know how many we had, perhaps 80 structures, something like that. But as we got more structures, we could look at different aspects in more detail. So one of the things that I looked at, and also Sauda Mini has looked at in detail, are the disulfide bridges, their geometry, where they occur, how close they can be along the chain. There are lots of different things one can look at. And actually, it was great fun doing this. There weren't many people around the world doing these analyses, were there? And you knew everybody. So it was a small group of people analysing these little motifs that you find in proteins that are great. They're detailed, but they're actually, when you look at them and you understand what the sequence preferences were, it's quite inspiring to look at it. And you began to understand more about this folding process. But you also began to see many structures with many folds. So you wanted to look at how two helices can pack together and how sheets can stack on top of each other and what the geometry is, because this is a well-defined physical chemistry problem. And there are, answers isn't the right word, but distributions that you find from looking at this packing. And of course, the last thing that became very apparent is that almost all proteins have these domain structures. And that if you have a big protein, it's rarely just one globular. It's made up by joining small domains together. So this concept of domains that I can't remember, frankly, when it was generally accepted that there were domains. But since then, it's been very clear that evolution occurs at the domain level, and that's very important to look at. And during the 80s, we got many, many more sequences. I must confess, I wasn't terribly interested in the sequences. I thought they were boring. They're just linear. Fact is, though, that they code, that they tell you about the structure. So from that point of view, I found them interesting. But this so the sequences led to the development of tools to look at evolution by looking at the sequences. This was quite important, and there are lots of different tools from different people. But the one I sort of know was by Des Higgins and Tony Gibson, Clustal W, and this program, I think, is amongst the 10 most highly cited scientific papers ever. So it's a really important contribution to the way that we look at things. And of course, with the realization of domains came the realization that we can have domain families. And people looked in detail at these different sequence families. But perhaps the most important observation, I think, came from Cyrus Chothia here with Arthur Lesk. When he was in the lab in Cambridge where they were looking in great detail at the hemoglobin sequences. And also they had lots of hemoglobin structures. And what they observed was that the sequence, if we're looking at myoglobin or hemoglobin, every single amino acid, apart from one, can mutate. That's the one, the histidine that binds the iron. That has to be a histidine. Everything else can change. But if you looked at the structures didn't really change fundamentally at all. So over these vast evolutionary times, different organisms from different branches of the tree of life maintain the same protein structures. And I still find that to be an amazing fact, that you conserve these structures over whole. So they're the same in plants as they are in animals, as they are in fungi, as they are across in bacteria. So you've conserved this geometry because the geometry determines the function, and in evolution that function rules. So this structure also, Cyrus led the inspection of the immunoglobulins, which of course are a very unusual class of proteins, because they are able, that family, are able to recognize any small ligand. In fact, any other protein. They can change, and they do it through these loops, and Cyrus noticed that there were certain classes. It wasn't a continuum. You only got certain classes of residues in there. So the conservation of structure and how that conservation, this plot shows as the residue sequence identity between a pair of proteins decreases, the structure gets more and more different. That said, these values are only at about 2 angstroms, which is very tiny. So they don't change very much, but they do change, and there is a relationship between sequence and structure in terms of the detail. Now, at that time, at the end of the 80s, horror of horrors, there were some structures that had been deposited in the protein data bank that proved to be incorrect. Now, if you do crystallography correctly, it's one of the rare parts of, well, biology, if you like, where there is a clear, this is right and this is wrong. And there are about 3 structures deposited in the PDB which they got wrong. One of them, they traced the chain in the wrong direction. So instead of going from N to C, the beginning to the C terminus, they'd put the C terminus at the beginning and gone backwards, if you like. And that was, people were horrified because that shouldn't have happened. It happened because people were trying to rush to get their papers out, basically. In some cases, people were just careless. Anyway, we thought it would, well, no, actually, this isn't why. I'll tell you why I started looking at all this stuff. This is the Ramachandran plot, which is color-coded according to the red parts of the most populated areas. What I was interested to see was the Ramachandran plot comes from consideration just of 2 peptides. It doesn't think about the broader problem. It's just looking locally. And yet these proteins fold into these complicated structures. And I was intrigued as to whether that folding would distort the Ramachandran plot. So we'd end up with something stretched because it was sort of taut. There were too many conflicting things within a structure. And so we plotted it. But what we found was that the clustering within these allowed areas depended very much on the resolution of the protein. So if you got a higher resolution protein, i.e. you're more confident that this is right, the higher it got, rather than the phi-psi values scattering across space, they actually clustered even more. So they obeyed the stereochemistry was even better in the higher resolution structures than it was in the lower resolution structures. And then we started to look at Chi-1 values. Now, this is real detail for those who aren't so familiar with structures, but the Chi-1 values are the side chain angle values, and those correlated beautifully with resolution, which is what you can see here. And we looked at a whole variety of things. We also, I should say, and this is important for data, we had a program that we called PDB Clean, because in those days, all this data on structures was deposited by hand, and people had to type in coordinates into the protein data bank. Now, there are about, perhaps in those days, the average length of a protein would be 200. There are 10 atoms in each in each side chain. So you'd have 2,000 times 3 for X, Y, Z. So you'd have 6,000 numbers to type in. And you made mistakes. People made mistakes. So the term Escherichia coli, there were 25 different spellings for Escherichia coli when we looked at the PDB in those days. So you had to sort of refine them. So we did this using a program, and that improved what we were trying to do. And, let's do it this way. Why isn't it working? Oh, okay. Yeah, it's all right. It's worked. So what we did was to develop something called protein, which was ProCheck, which validated these structures according to their stereochemistry. And this shows, we developed ProCheck. The program actually was made available in 1993. And then it was based on the Ramachandran plot. That was what I wanted to say. It was based on this original observation. And in fact, these phi-psi angles are the best way I know to check that the stereochemistry in the protein is good, because you can't fiddle them. Because if you change one phi-psi, that affects the rest of the chain. And so you're distorting it. With chi1 values, you can just twiddle and use the side chain libraries, but you can't do that with the Ramachandran. And so this Ramachandran proved to be absolutely... So this plot was based on the structures available in 1991. That was what we did. Since then, other validation programs have been developed, and what you can see with these other programs is that gradually, actually, rather than getting less clustered, as we've got better data and better methods, these cluster even more into Ramachandran's favourite areas. So that initial observation really has been critical for assessing the quality of protein structures. So at this time, we began to get lots and lots of structures. And so we had to, and lots of sequences, and those had to be compared. Now I suspect the students here are all familiar with BLAST. Have you all done BLAST? Yeah, you've all used BLAST or something like BLAST. So there were lots of different programs that tried to do this. I myself wrote a structure comparison that when I was in Oxford, called SUPER. So I had SUPER for superpose, then I had SUPER A, that was the next version of, then I had SUPER B, and I went to a meeting and somebody said, I've been using the program Superb, which wasn't the idea at all. It was just, I was trying to improve my structure comparison. But in fact, it wasn't a very good tool. But Willie Taylor developed an excellent tool called SAP that, in fact, with Christina Rengo, and we use that as the basis of CATH, which I'll come on to. But they're also visualizing the multiple sequence alignments, and Jeff Barton, who was also at Birkbeck with Minnie and myself, he's developed a very nice package that looks at these multiple sequence alignments. So all these different tools and databases are used. I added in here FoldSeq from Steinegger, which was developed, 2 years ago now. And that has really changed our ability to compare structures quickly and accurately. And so these developments, these programs were done in the 80s and 90s, but this recent development, has made the comparison of structures even much, much quicker, which is what we need now. So the term bioinformatics didn't actually appear until the 1993. And I am often told by my genome colleagues that this came when people started looking at sequences. Not right. It's the structural world for a long time had used these methods of looking at structures. And you can also see that the number of publications rapidly increased and has increased hugely since then. But as we got more structures, another thing that happened was that people would determine a structure and publish it in a paper, and then somebody would write and say, your structure looks just the same as mine. And they didn't know because the sequences were so different. And so what you see here when we were doing our comparisons, and this is Christine, my colleague at UCL, this just shows 4 structures where the core of the structure is well conserved. In white, you have the sequence similarity between the 4 structures. They're 4 enzymes. in green, you have the sort of measure of structural similarity. So although the sequence changes, really, there are very few sequence amino acids conserved, but the structure is conserved. What was even more disconcerting to me as a non-biologist was that these 4 proteins, which are clearly related when you look at them in detail, if you look at their names, you would never know that they belong to the same family. And that was extremely worrying, because I'd always assumed that cytochromes look like cytochromes. In fact, cytochrome B5 is completely different from cytochrome C. And so we needed these quantitative methods of comparing these structures. And that relates. And so this then, we decided that we would create this domain database, CATH, Class Architecture Topology Homologous Superfamily, in the 90s, and Christine was my postdoc at that time. Simultaneously, in Cambridge, Cyrus Chothia and Alexei Mertzin developed the SCOP database. And basically we would do it, the goal was the same. We were creating an overview of the world of protein structures. The methods were different. Alexei is an amazing person who can look at a structure and know which family it belongs to. I'm afraid that's beyond me completely. Now, we used computational methods to try and do this, which in some ways is better, in some ways is actually worse, because sometimes these things have evolved so far that you can't tell the members of the same family. But Alexei could see the differences. So we developed these 2 data resources, CATH and SCOP. And in fact, Minnie also developed a data resource here based on the work when she'd been in Tom Lundell's lab and carrying on from there. And this really is our human way of trying to understand the universe of protein structures. It's a way to look at it and try to comprehend what's possible and which are related, which are homologous. And in fact, that SCOP and CATH are working together to try and bring this together and to do it through PDB at EBI. But as I said, this idea that actually these new structures sometimes look like old ones. So we decided, well, this was my personal experience. I had a new student, Mark Swindells, who was work on a case studentship with Pfizer. That means that it was with a commercial company that were funding it. And they wanted to look at interleukin-1 beta. Now, the structure had been published, but the coordinates weren't available. But it had the same structure as soybean trypsin inhibitor, for which the coordinates were available. So I said to Mark, well, never mind, just go and do the sequence alignment, and then we'll be able to build a model of interleukin-1 beta. And poor Mark came back a week or 2 later and said, I just can't get these sequences to align. They're just completely different. And they were 9.5% identical. And that was why he couldn't do it. And so this idea of recognizing folds came into being. And David Jones, who was in the lab at the time as a student, he went on to do fold recognition or threading, trying to recognize the fold from the sequence. But you can see here, at that time, there was a whole slew of new development of methods and observations. Molecular dynamics and the simulations were developed by Karplus in particular, and Levitt and Berenson and Van Gunsteren. There was normal mode analysis. There were a lot of detailed electrostatic field calculations from Warwicker in Manchester and Barry Honig in the US. There were a lot of methods on ligand docking. Manfred Sippel was a proponent of using reverse Boltzmann calculations to develop force fields based on proteins. And we certainly used that. I thought that might solve the problem of protein structure prediction. Sadly, it didn't, because there's not much difference. At that time also, Alfonso Valencia, now in Barcelona, observed the covariance of close amino acids, which actually we'd observed and other people had observed, but it was documented in a paper, and this has later been one of the most important contributions to solving the protein structure prediction problem. But David worked on fold recognition, and again, CASP. So this is every 2 years there was a meeting that was called CASP, and it's the comparative assessment of structure prediction. Some people call it the competitive assessment of structure prediction, because of those in the field know this became quite a competition. So the idea was every 2 years the organising committee distributed the sequences of a set of proteins that were in the process of structure determination. And then the predictors had to do a blind prediction to predict what the structure looked like. The structure came out, and then you could compare the predictions. And the reason this happened, in part, was because not many, but some people claimed to have solved the protein folding problem, whereas in fact they hadn't. And once this came about, you couldn't pretend. There was this regular 2-year assessment whether you'd got it right or wrong. And so John Moult in the States, funded by them, set up this 2-yearly competition, and they got external reviewers in to assess how good the predictions were. So I was one of the first ones that did it. It was a huge amount of work, actually, even then. But that was early, and many of the structures that were deposited by the labs had lousy stereochemistry. So we could check it with ProCheck and say, they had phenylalanines that crossed through each other and also that sort of thing. It was just bad. So this improved, without a doubt, the things. At the same time, the idea of structural genomics emerged, which maybe you guys out there won't know what that was. The concept was we did because genomes had been determined. So we had the whole human genome. So why can't we have the whole human proteome and know all of the proteins? And that includes many proteins, even now, whose functions we don't know. So the idea was just blindly to go and get a sequence, solve its structure. And there were the efforts across the world in this structural genomics. The problem was we didn't know what the function was, and so biologically this was not terribly helpful. But what did happen as a consequence of this, that the methods of solving protein structures became much better, expression became much better, and the whole automation of solving protein structures improved. But this highlighted the need for us to go from structure to function. And it was at this point that I decided I didn't have any more ideas about improving protein structure prediction. It was just too difficult. So we turned our attention to looking at functions, which, as a physicist, the first 10 or 20 years, I wasn't really interested in what the proteins did. I was just interested in what they looked like and the sequence structure. So this change to function was quite a challenge. And so at that time, there were lots of methods to look at different interactions. So protein-protein, because function almost always involves interaction, either with another protein or with a small molecule ligand or with DNA, RNA, or with sugars. So we did a whole lot of analyses on these interactions, as did many other people around the world. And this just, to me, highlights some of what we found. So this is let's look at the bottom one. This is looking at nucleotide adenine dinucleotide, NAD, in 5 different protein structures. And at first, people thought, well, if you're binding NAD, the pocket in one would be the same as a pocket in another, the same as a pocket in another. In fact, that doesn't work, because you can see from here, if we just look at the NAD to begin with, it's flexible. So it's got different conformations. So the shape of the pocket is going to be different, full stop. But even when it's extended, actually you get different shaped pockets. There are many ways to recognize one small molecule. There isn't just one way, one shape of a pocket, one feel thing. So that was, but just gathering this information has proved to be in itself demanding, with the development of many new methods. The functional analysis, oh gosh, I'll have to go quicker. The functional analysis was helped by the introduction of the Go annotations. Genome ontology, it's called, and this was there were a lot of people involved. Michael Ashburn, as shown here, who sadly died, he developed this Go annotations. And when he told me he was going to do it, I thought, that's impossible. That's just impossible. Capturing function in 3 descriptors, basically. But I didn't reckon with Michael, who knew more about biology than than anybody, because of his studies on Drosophila, actually. And so he developed this, and this really helped us to understand more about function. I just mentioned that the transcription factors and how they work and control expression has been also, the control of biology has been a central theme. So over in the 2000s, there was a lot of work on networks, data integration, many, many more structures, docking, the emergence of the fact that about a third of residues in proteins are disordered. What's the impact of that on function? And that's important and is being worked on here and elsewhere. Function prediction from structure, and they developed something called CAFA, which is comparative assessment of function annotation, the same as CASP, and that's ongoing. But there were many, many more structures, and despite all those structures, there were even more sequences. And so only a very small percentage of uniprots could be directly mapped onto the PDB. Tiny. And so the challenge of protein structure prediction became even more important, because we got so many sequences there that we wanted to look at. So in the 2000s, 2010s, the way to predict structure was basically using homology. And because of the conservation of structure, building one protein from knowledge of a close relative, and there are now, this was done by hand originally, but then the development of many new, very, very robust methods for doing this homology modelling were developed across the world. Charlene Blunday, Thorsten Schrader, David Baker and co, and Mike Sternberg. And there are other programs as well. But a breakthrough came in CASP14 in 2018, and here's John Malt. And this just shows the quality of the predictions. Along the bottom, we've got all the groups that were involved in this meeting, CASP14, and we've got the number, well, here it's a measure of a Z-score, actually, of how the different groups did. Now, normally, in every previous CASP, this has been sort of a continuum like this. This was the first time that one group had stood out way above the others. And that was AlphaFold 2. And it was done primarily by people who were not structural biologists. They weren't physicists. They were computer scientists who developed different AI approaches to solve this problem. And here you can see where this is a measure. So each CASP, there would be a graph produced about how good it was. Similar proteins were always predicted quite well. The difficult the predictions for the difficult, very distant proteins were rubbish. But gradually, this curve moved up, but very, very slowly. And it wasn't until AI methods were introduced here in CASP 13, but then in CASP 14, what these little circles show is that for, I think, 96% of the targets that were predicted, AlphaFold got the most accurate structure. So this was clearly a step jump in the accuracy. And this has changed things really tremendously. The structures, some of them built like this one, look very good. The only differences are loops that change a bit. Some of them are really, they just show you that you've got a flexible protein. Others are a mixture between a core that's defined and loops that aren't defined. And so it depends on the protein that you're looking at, what sort of a model you get out. It was very clear right at the beginning that you got better models out when you had a relative than if you don't have a relative. So if you have a close relative, the quality of the models, the confidence score, that this PLDDT score shows that for the homologs, the average PLDDT score, the confidence was much higher on average than it was for proteins without a structure. So protein structure prediction really is the perfect problem for AI. And the reasons are shown here. It's a long-standing problem. There's lots and lots of data that's curated, clean, accurate, freely available. Good metrics to measure success. And of course, evolution, which determines what these structures look like, means that many of these structures have relatives. And so that helps in the AI prediction. So over the last 30 years until AlphaFold was developed, there were many useful technical developments. But this, effectively the AI brings together all those developments and treats it purely, if you like, as a technical mathematical problem to combine lots of data from lots of structures to make your best prediction go forward. I think I might just skip some of this because just to say that the AlphaFold database, so initially we had hardly few, we now have 214 million structures predicted by AlphaFold that are freely available. Anybody, you can go type in a protein and from the EBI you can pull down the predicted structure. This has been phenomenally successful, and you can see there are over 2.5 million users of this database since it was set up. And it's across the world. You can see here, this is the number of users, and you can see in India there's quite a lot of use compared with many, many other countries. Of course, as always, America and China have the most use. These structures are used not just for structural biologists, but because they're available for every structure in one shape or form. They might not be good, but they're there. Then in the clinical sciences, genetics, microbiology, chemistry, biochemistry and cell biology, and of course in bioinformatics. So it has wide usage across here. But, just as a caution, people who are using these structures aren't structural biologists, and they really do need to be trained about what the models mean, where are the good bits and the bad bits, and how to use them. Oh, sorry. And there is now online AlphaFold, a practical guide that's been developed to help people use this and to use it well. So I encourage, if you're interested, you can go and look at this and get a sort of a tutorial on how to use these structures. So now we've got all these structures, what can we use them for? We had 200,000 before, now we've got millions. And of course, perhaps the most important thing is we can interpret ligand binding sites, variants, mutations, multimeric states, etc. for proteins. But we can also, as well as providing this structural context, we can predict multiple conformational states in various ways. We can compare and contrast structures. We can predict protein-protein interactions and identify very distantly related proteins. And one way this is important is if we're looking, in this case, at bioremediation, where you're trying to use biology to clean up waste dumps, etc., you can actually identify that you want to nitrolyze enzymes. And you can go and search in Magnify, which has these 2.5 billion sequences for nitrolyses that might be useful for bioremediation. So it gives you proteins that you know are extant that can really help in doing this. So drug discovery, tackling neglected diseases, understanding antibiotic resistance, fighting plastic pollution, increasing the health of honeybees, I rather like that, and understanding how ice form are just some of the examples of that it's been used to. But the example I like best, inevitably, is the one that reveals the universe of protein structures. And this is published by my old colleagues, Christine and David, in their groups at UCL. And it's called the Encyclopedia of Domains. And what it did was to go through these the high-quality structures, 14 million, and cluster them into the Cath database that we developed before to see what they are. And what they couldn't use all of them because some of them aren't good quality, but they were able to put 90% of those structures into known Cath folds. And in total, they got 7,500 new folds. And here you can see some of the new folds. These are the symmetric ones, the pretty ones. They look really beautiful. And so this approach learns well enough to predict novel structures as well as predicting structures for which there are also relatives available. So that, I think, is quite important. They also showed that certain families of proteins have expanded enormously over the different organisms, and it's not equal. Some families don't expand, others really expand. And so where does the future take us? So there is for structural biologists, there is a new normal. You now probably have a prediction for your structure before you ever start to solve the structure. And so that gives you some insight into what you might do to do that. I should say there are obviously many more databases. So when I joined EBI in 2001, we only had 5 data resources. We now have 40 just at the EBI. There are many new specialist databases in different topics, and there will be more and more going forward. Obviously, for structures, we need new databases. So we need not only the PDB, but an electron microscope database and a public image archive that I think are very important in structural biology. And really, we're going from structures of individual molecules to creating structures of these very large complexes. This is a nuclear pore. And you can make this structure, you can model. And again, here in NCSB, there are, people doing this. So this is really, I think, the future in a sense of structural biology. But not only that, we can actually see with tomograms through single cells. So you can see the individual components of a cell, and then you can even pick out individual the big molecules like ribosome, but gradually I think that will increase. So we will, I hope, not just know about the universe of structures, but we'll be able to see in the cells exactly where those structures are and how they interact together. And I think that will be a big difference. And here you can see in situ structural biology of mitochondria, and there are various blobs like a chaperone molecule here, an ATP synthase here, but here's a blob of a structure that they didn't know what it was. So we learn new things by these tomograms, I think. So where do we go? So this is building model biology at different scales, which is, and I don't have time to go into that. But I just wanted to finish with saying what impact will it have on the wider community. So we know that there are many national genome projects. I put this slide together last year. There are 220 different genome projects worldwide. These are generating genomes at a great rate. This is going to change as we have this data, our understanding of genomics and biology, without a doubt. These proteins we know we'll be able to use in these very different areas: sustainable bioenergy, biodiversity. There's somebody doing biodiversity of tigers in NCSB, which is fantastic. You know, how much do they vary? Biotechnology, engineering enzymes, which would be fantastic. But to me, the greatest potential, in a way, lies in the medical sciences. And in a way, it's a really good area for AI. There's lots of data. There are many expert clinicians. It's a highly regulated profession. And the current approach is mainly based on previous experience of the clinicians. And I should say the imaging is going to be really important. But the problem is in the quality of the data, which is still not there. There's lots of it. There are all different sorts of data that you can see here. In general, it isn't very structured and organized. The biobank and cohorts are the exception here. But generally, hospital data is not well structured. And so there's a huge challenge in the next 10 years, I think, to build the data infrastructure for medicine, for medical data. In our world of structures and biology, it's taken 20 years to build up this ecosystem of different databases. The medical problem is even more challenging from many different aspects. The sharing data, the policy and the ethics for that, the data mining, the data security and regulations and crossing borders. Nearly all medical data is in the language, the local language. You know, we don't have that problem with protein structures, thank goodness. It's all the same language. But this is a major challenge. And in fact, in the UK, we've set up Health Data Research UK, and their mission is really not to do the research, but to facilitate finding, accessing, linking, curating, analysing, and getting new insights, and getting those into the clinic to improve health. And this is, it's about 3 years old now, and it's a huge problem that's faced in the UK. Even though we have a National Health Service, and it's not commercial, even so, it's a real problem to do this. And this is one of the major challenges where I think AI will come into its own. So I'd just like to finish by saying, to date, molecular biology, structural biology, cellular biology, developmental biology, organismal biology, ecology, evolution, the medical stuff has all been in its own silos to some extent. It's not been as integrated as it needs to be. In my mind, the only way to do this integration is using bioinformatics tools. It is the only way. There's no other way to bring this information together. So the emergence of bioinformatics, it was peripheral at one stage to most biologists. That has to change. And it will change in terms of building models and going forward and really interpreting this huge amount of data that we've got. And now we are much closer to the medical application than we were 50 years, infinitely closer. There was no link in those days. And so the idea of improving drug design, treating health, and generally improving our lives and maybe helping to tackle climate change in some way. So what we'd like are better methods, better biology, better medicine, and I'm sure you've heard about healthy planets, healthy people, and healthy planet. And all these new technologies can be brought together using bioinformatics methods. So I just close by thanking the Foundation again for letting me come here and speak, but also all the members of my group. We got together last year and had a wonderful get-together of students over many, many years. And it was really nice to see everybody again. But thank you to them for all the work that they've done that has made my life both a joy, sometimes a disaster, but mostly a joy, and for helping us to understand the challenge of proteins and protein structure. Thank you.
Wow. That was a very beautiful articulation of not only the history of bioinformatics, but also to say how bioinformatics is expanding and the horizons are much more than we think. So to begin with, we would like to ask if there are any questions from this hall. Questions? Okay, maybe I will... Okay, Rana. Janet, so the AlphaFold, if we have a query sequence...
Sorry?
Using AlphaFold, if we have a query sequence and it connects to two domains
yes.
Of known structure, but it is discontinuous, do you think AlphaFold will have a problem? Or
I don't know the answer to that. That will be very interesting. And of course the answer is probably be if it's an ab initio sequence, I don't think it would solve it.
Okay.
But if there's already a structure there that's got domain swaps in it, then it would.
Thank you.
So I think it depends.
Hi, Janet. Thanks for a wonderful talk. You said many, many interesting things. I just want to ask about one of those. Is it possible to get a alpha dynamics of like a conformational dynamics for like that? And where is the training set for that? Because we haven't built that.
Well, I think that is exactly the problem. I know that John Jumper is really, his real interest is dynamics. And so I think we will see that. But with all this AI, which I stress for the medical data, it's getting the data to train the programs. And I think maybe we can learn how the dynamics is structured. But I don't know. I've always been so worried about whether the dynamics is accurate or not that I've tended to avoid it if I possibly could, because it's so difficult. And I spent my PhD developing force fields and all sorts of things, and said I would never again do energy calculations. But actually, there's no option. That's the only way to really look at it and look at these dynamic things. So I think we need, but there are some things where dynamics matters. There really are some aspects of function where you don't need to consider dynamics. And so a parsimonious approach is always the best one, where you do only what's necessary, I think.
Hello, ma'am. First, I would like to thank for your informative speech. So my question is like this. How do you think protein protection helpful regards human health and diseases?
Sorry, you'll have to say that more slowly.
Maybe I can repeat. How do you think structure prediction can help with human health and diseases?
Oh, health and diseases. Yeah. So that depends on the first thing, it helps us to understand how things work. So if you think about a car and it goes wrong, if you don't know anything about the engine and how it works, how do you begin to put it right? Now, it's not a terribly good analogy because a car is rigid, it doesn't move, and it's just a car. Human body, we've got 20,000 genes with all their variants and post-translational modifications, and we don't know how it all works, and it's very difficult to model it together. But if we don't have the structure, very often we can't say anything about how that protein works. And so just, I was talking to the people from IMSTEM, and they're looking at sickle cell disease. There's one mutation in general that causes that sickling. But it causes the disease because that little red blood cell, in part, can't get through the capillaries. So it's the structure of the protein distorts the ability to bind the iron, which distorts the shape of the red cell, and it can't get through the capillaries. Now, that's systems biology. It's going right from an individual molecule with an individual mutation to an effect on the cell that in turn affects the biology of the person, the health of the person. And that's just one of our 20,000 proteins. For many of, even with human proteins, we don't really know the detailed function of many of those human proteins yet. So we need to know their structures, but that's not enough. We need their structures, we need their expression, we need to look at it in situ, in vivo. So yes, to me, the structures are an absolute sine qua non. You need them for understanding. From that, we can then develop, with all the other data and knowledge about what goes wrong with health, in bad health, and hope to find ways to make it better. So it's still a long way for many diseases, especially the chronic diseases that occur over a lifetime. But there are lovely examples, of course, where given mutations cause given effects that for development for children, and you can give these children a drug that makes them okay. But those are frankly the exceptions rather than the rule. So we need to understand before we understand. And that's what, as researchers, that's what we all do. We try and understand the problem. So I don't know if that answers it. So you're not going to be healthy tomorrow, but in your lifetimes, you guys, one, I'm sure you will have your genome sequences done. So you will know every mutation that you've got. That may be good news or bad news, and it's not clear exactly what they should tell you. But medicine will change, I'm absolutely confident, because of the data, because of our understanding, and because of the AI methods that I hope will help the doctors to predict what's going to happen and to recommend the best medicine in the right doses. So there's lots and lots of things to do. Okay? You don't look convinced, but...
Over there.
Hello, ma'am. Thank you for your talk. Actually, I have a doubt, what is your opinion about predicting the active site of a protein structure? There are many online tools are available to predict the active site, right? Like Cas2b. So how reliable is that to predict the active site of a protein structure?
To predict the active, you're asking several questions there. Predicting the active site, so active site to me implies an enzyme, so let's think about enzymes first. Predicting the active site of enzymes is actually not too difficult. And it's not difficult because the catalytic residues are generally absolutely conserved. So you get this patch of very high conservation in the protein where the active site is. So that's not so difficult. Predicting the effect of a mutation is more complicated. Those mutations actually are rarely in the catalytic residues. They're much more commonly in the binding residues. And their effects are difficult to predict. So you can think of it as the phenotype of the protein. If you have a mutation, how does that change the behavior of the protein? And there are many programs that do that. With the structure, the first thing to ask if you have a mutation is, where is it in the protein? Is it on the surface? Is it buried in the core? Is it in a cleft, etc.? So having a predicted structure, or even better, an experimental structure, allows you to have some insight into that. But it depends exactly what question you're asking. But it doesn't solve, having a structure doesn't solve some of the problems. I was thinking, some years ago we had a foot and mouth disease outbreak in the UK when they had to slaughter a lot of the animals. I mean, it's just horrendous. We've known the structure of foot and mouth virus for at least 10 years before that happened, and yet we haven't solved the problem. So a structure is the beginning, it's not the solution in itself.
Thank you.
Right. Are there any questions from the other lecture hall? Oh, there is one question over there.
It was a very good talk, ma'am. By the way, ma'am, myself, Vinay, and I was working on nanotechnology, nanoparticles. Is there any websites where sequence could be sequenced and materials, extract or any nanoparticles can be integrated through that, like with our structure and protein structure? Can we see the interactions?
Sorry, I'm not sure I
think
understand.
the question is, given a protein sequence, can we predict whether it will interact with nanoparticles?
Oh, with nanoparticles. It depends a bit on the nature of the nanoparticles and the nature of the protein. In terms of detailed interaction, I don't know. Has anybody actually tried to do that?
Yes, ma'am. Like through the literature survey, we have seen that even a DNA can be interacted with nanoparticles. So how is that proteins could be interacted with nanoparticles?
Oh yes, proteins will interact with anything that's around, basically. But they won't necessarily interact specifically.
So if we know that structures,
perhaps
it could
can continue discussions over the tea, please.
Yeah.
Yeah, very nice. So there, I must say that this lecture is also being streamed in another hall. So are there any questions from the other lecture hall? No. Any questions on the Zoom? No? Okay.
So we... Okay,
with this, perhaps then we should please join me in thanking Professor Janet for a very nice lecture. Yeah.
Thank you.
Great. So I would request our Center Director, Professor Shashi Dara, to give a small memento to the speaker, please. We want to say this one there, this one. I would also request Mrs. Asha of the Shivalal Philanthropic Initiative to give a small memento to the speaker, please. Yeah, I must also acknowledge the fact that this whole public lecture was co-hosted by the Indian Academy of Sciences, Bangalore. So I would request all of you to give a big hand for organizing the students from different parts of the city today. So with this, we should conclude, and I would request that you all can have high tea, which is kept just outside. You can please join us. And also that there will be another public lecture tomorrow in the Indian Institute of Science, same time, 4 p.m. It's on enzymes, evolution, and diseases by the same speaker. Yeah, it will be in the Biological Sciences Auditorium in the Indian Institute of Science. You may join there as well. But now please join us for high tea. Thank you.